Welcome to episode 6 of the Sustaining Soils podcast, brought to you by Valent Biosciences. I'm your host, Lisa Peterson, and I'm excited for our episode today as we discuss innovative partnerships and cutting-edge technology in plant science and imaging. Our guests today are Dr. Christopher Topp and Keith Duncan from the Donald Danforth Plant Science Center in St. Louis, Missouri. Chris grew up in Chino, California, and pursued his undergrad through a PhD in plant biology at the University of Georgia. After launching his professional career at Duke University, Chris is today a principal investigator and member at the Danforth Center. For almost 20 years, Chris has studied fundamental processes, both small and large, that drive the growth and productivity of crop plants. His team at the Danforth Center continues to develop technologies and infrastructure that enable the analysis of the hidden half of plants. Welcome, Chris.
Glad to be here, Lisa.
We are so glad to have you. And we're also glad to introduce one of your other team members in your lab as well. It is my pleasure to introduce Keith Duncan to our listeners. Keith is a research scientist in Chris's lab at the Danforth Center, and he is also the director of the X-ray imaging facility. Keith completed his undergraduate degree in plant biology and attended grad school in plant pathology at the University of Georgia. His professional career began when he joined Dr. Rick Howard's lab at the DuPont Experimental Station using light, laser, and electron microscopy to study plant microbe interactions. He joined Chris's lab 8 years ago and also added a master's degree in science education from the University of Delaware. We're so glad to have you with us as well, Keith.
Thanks, Lisa. It's good to be here.
Well, I am very excited for our conversation today and to be able to dig in deeper to what the Danforth Center does and specifically what you two are doing within the lab. So let's go ahead and get started and just give us a little bit more of an overview, Chris, of the Danforth Center and the research that it is doing within the agricultural industry.
Sure, I'd be more than happy to. Although this isn't an easy question to answer. One of the incredible things about the Danforth Center is the breadth and diversity of plant science expertise we have here and our ability to ask and answer a broad range of questions. And actually, there's research here happening from the very basic level up to very applied work. So one of the most notable recent examples is actually international work developing virus-resistant cassava plants for sub-Saharan Africa. And this has just been an incredible scientific achievement. I think you mentioned I've been here about 10 years and I've seen not the entire, but much of the arc of this. And today there are virus-resistant cassava germplasm being disseminated throughout sub-Saharan Africa, and it's just made a huge difference. So these are the type of big and long-term problems the center can work on. There's also basic research happening here, and this is important. Some of your listeners may have heard just in the popular media or anecdotally of CRISPR, technology. And so we have researchers here. In fact, our president is a virologist who worked on these really fundamental interactions of viruses, plants, bacteria, and it's this type of basic research that led to the ability for us to do genome editing and to employ CRISPR to develop more sustainable, resilient crops and crop systems.
Phenomenal. So it sounds like there is a lot going on and a lot that is adding to the overall advancement of the ag industry in multiple places, not just here in the U.S.
Indeed.
Keith, why were you drawn to the Danforth Center? I think you came, about 8 years ago, you joined Chris's lab?
Yes, it was about 8 years ago, in fact, this August. And the story of how I got here is really unusual because about 8 years ago, the industry was consolidating. So the very large chemical company based in Delaware, where I worked, decided to merge with a very large chemical company based in Michigan. So as soon as they merged, in order to get through the SEC, they had to break themselves up into many smaller bits. So after 25 years, in this particular research position, I was suddenly laid off. So we knew there was nothing left in Delaware. We knew we had to leave. So we looked around the country to say, where do you go to study plant science? Where could I have a job in the plant science industry? Where's my best chance? And without question, it's St. Louis. St. Louis is really the center for plant science research in the country. So we rolled the dice. We just moved to St. Louis sight unseen and hoped to find work. And by good fortune, Chris was in the middle of taking delivery of a very large X-ray tomography instrument, which we'll get into shortly, and was looking for someone to run it. So here is a plant imaging specialist who just turns up into St. Louis right when Chris puts out a job ad. So tremendous good fortune, and it's just exploded from there. We've both been really lucky to have ended up in that situation.
What a neat story of how that worked out and what brought you to Chris's lab. So speaking of Chris's lab, Chris, tell us a little bit more about, what your research focus is in your lab. And in your bio, we mentioned that hidden half. What do you mean by that?
Well, the hidden half refers to the root system and the impacts they have on the soil and the microbial ecology. And we call it the hidden half for an obvious reason. It's also the largely ignored half. And so that's really the purpose of my research program, is to shed some light on a really fundamental, foundational part of plant and crop health and productivity, of soil health. But to do that, we had to develop, and are continuing to develop, cutting edge methods to do scientific research on roots and their interactions with the soil. And that's really was the motivation for bringing this 8-ton piece of equipment and then a subsequent multi-ton second piece of equipment, which is another X-ray system that allows us to actually image microbes and things at a very small scale. And it was incredibly fortuitous to, I can't say recruit Keith. It was just a absolute random chance in the universe is what it felt like. And in fact, it almost didn't happen, because when I got this email from Keith, it seemed a little good to be true. So I'm wondering what's the backstory on this person who seemed a little overqualified for the position. And so it took me a couple of days to get back to him. But then once we met, we quickly realized that this was our opportunity. And Keith, I think your first day was the day the X-ray imaging system was turned on. So you've been there developing it the entire time.
That's right.
That's right. Yeah. And I just want to mention that this was an incredible example of what industry and academic research collaborations can do. I had just received a small, well, it was not a small, but I had just received an NSF grant to work on nitrogen and root relations in corn. And I had proposed actually to build sort of a homemade X-ray system. So I was pricing out leaded drywall and
things you never thought you'd have to know as a scientist, right?
Exactly. Things that even the Home Depot doesn't have in ready supply. And so, and along comes Valent, and there's an incredible story here. I don't think we can tell it all here, but just with the incredible support and vision of Valent Biosciences, and Warren Schaefer in particular, we were able to acquire additional support from them that allowed us to get this instrument, which was at the time the only one in the public sector in the United States. And this X-ray system allows us literally to see the hidden half of plants, and we've been using it for that ever since.
So let me back up and get this straight. So about 8 years ago, Valent Biosciences was the industry partner. You got the grant from the NSF, and the 8-ton CT X-ray machine was bought. Am I correct on that?
That's right. Yeah, that's right. The only part you're missing is the part where we had to cut a hole in the side of the foundation of the Danforth Center to get the instrument in the door. That took 3 or 4 days, and it was a little dicey, but we did it.
That sounds like a little dicey. So Keith, you're the one running this, correct? You're the director of the X-ray kind of imaging side of things. Tell me more. We've talked about this machine a couple times here and alluded to this 8-ton. Tell me more about it.
And that's a great question, Lisa. This X-ray tomography instrument is very similar to the kinds of things that you'll have in a hospital, where a human patient will go in and the doctors can use what they call a CAT scan. That's an old-fashioned name for computer-assisted tomography. They'll do a CAT scan to look inside the human without cutting them open. Well, these instruments, they also make industrial scale ones, and very large aerospace and automotive companies will have a dozen of these type instruments to do non-destructive testing of their various parts, looking at piston rods or engine blocks or other parts, other components that need to be inspected before they can be used in an airplane or a car. And that's where Chris was really very clever in choosing this instrument, because the big industrial instruments are, well, they're big. They have a large internal cabinet. They can hold heavier weights, and they have a lot of versatility in terms of how many different kinds of sample sizes you can look at. And by look at, I mean non-destructively look into whatever it is we put there. So we can grow, for example, corn plants in various pot sizes, put the entire plant in there, and use the X-ray to look through the soil, and then be able to, using the computer, actually separate out the root system digitally instead of having to dig it up out of the ground. So, as Chris said, we're studying the hidden half, and we need to be able to literally look at the root system if we want to see, is it branching more? Is it branching less? How does it occupy 3D space? Because they're such important traits for breeders to make sure that the plants are doing the best they can with the resources they're given. So that's a huge aspect of what we're using X-ray tomography for. How can we look into plants in a non-destructive way without having to rip them up out of the ground?
Absolutely fascinating to me. And every agronomist or farmer or scientist listening to us today is jealous, because anybody that's had to dig roots out in the cornfield knows how much of a challenge that is, trying to figure out what's going on in the hidden half. So what fascinating work. So Keith, in follow up to that, you've also added another piece of important equipment. Tell me about it.
The X-ray instrument we have, the large format instrument, is great for looking at large samples. But what we were able to do is also leverage our business partnership with Valent Biosciences to add an additional X-ray instrument, which is an X-ray microscope. So between the two instruments, we can look at a wide range of sample sizes, from an entire corn plant all the way down to microbes in the soil. So what this has allowed us to do is expand not only looking at the hidden half of plants, but looking at that entire space that roots occupy. We're looking at the roots and the soil and their interaction with the microbes, and exactly how that soil is made up. Soils come in an almost endless variety, and how they occupy 3D space is also very important to understand, because soils in different parts of the country are going to be made up of different materials, and being able to look at and measure and evaluate soil is almost as important as the plant itself. So that whole environment, that whole world below the soil line, is just now available to us to study.
Chris, as a follow-up to that, how has this technology enhanced or changed the research that you're doing within your lab?
Well, it's fundamentally changed it. So we discussed some of the difficulties in studying the hidden half. And traditionally, there's really two camps that are far apart. There's groups that study roots in the laboratory on very artificial media, such as petri dishes and transparent agar, things like this. And then there's, of course, digging roots up in the field and trying to make sense of them, with a pinboard or a rating system or something, using our human judgment and pattern recognition. And the two really have had a hard time meeting one another. And so the fundamental discoveries in the laboratory have rarely translated to the field. And we've not learned enough about some of the mechanisms of roots and their interactions with soil to achieve the goal of more sustainable, resilient, and productive agriculture by focusing on the roots. And that's really what we're here to do. We recognize today that we, of course, need to continue to feed people and increase the amount of food that we make, but we have to do it with fewer inputs. We have to do it in the face of more severe weather patterns. Mm-hmm. And somehow we have to do both hard things at the same time. And so what the X-rays allowed us to do is make that bridge between what we can learn in the field, which is directly relevant to agriculture, but in many cases is very information poor. It's very sparse. What we can learn from digging and looking at a root, even many roots, it really limits the science. And vice versa, most things that work well in the lab, when they get to the field, they do not work. And what we've been able to do, and we do all of these things, okay, we dig a lot of roots in the field, and we also do lots of work in controlled environment. And the X-ray system fits right in the middle. It allows us to evaluate. I think later in this podcast, we will have the opportunity to discuss some of those successes where we move things from basic science into roots in the ground, in the field, that we have real hope can help develop more sustainable agricultural systems.
I'd love to go there now. How do we bridge that gap, and how have you been able to bridge that gap from the research to the practical application in the field?
There's a few premises that we have in the lab that we start with. And one is based on the recognition that roots have been ignored through the process of breeding for yield under high inputs. And we've learned that in many ways roots have improved over time through this indirect selection, but they haven't improved to their maximum capacity. For example, approximately 50% of the nitrogen inputs in United States and really global agriculture are not captured by the crop. And there's many reasons for that, but one of them is that the root systems are not in the right place at the right time. Root systems and their allies, like arbuscular mycorrhizal fungi, for example, which also can help capture this nitrogen, are not in the right place at the right time. And this obviously is an incredible waste to the grower. It's a waste of money. It's a waste of time. And it has real significant environmental consequences. One example is nitrous oxide, for which agriculture is one of the largest man-made sources. And much of this comes from nitrogen that's not captured by the crop. So we figured if we could move the needle there, that would have a lot of benefits. We could potentially use this to both improve the productivity and the sustainability of our cropping systems. And so I don't want to get too nerdy and technical here, but my background is actually in plant genetics, and I'm a plant scientist, and that's what I'm trying to bridge that gap into agronomy and real agricultural systems. And so the X-ray has allowed us to evaluate some of our basic research. We've identified genes that control the root system architecture of corn and other crop plants. And we've shown that, well, this works in the lab. And we move it to a greenhouse and we evaluate it in a soil with the X-ray system. Hey, it still does what it did in the lab. So let's put this out in the field. And we now have multi-year long-term experiments in the field that show that we can change the root system architecture of corn plants. And then we're measuring the benefits of this. We're measuring, of course, the grain, amount of grain, that's nitrogen capacity. We're also measuring soil health parameters. We're measuring, we can see reductions in nitrous oxide emissions. We can see accumulations of increased aggregate stability. We think we'll see increases in soil carbon, and water infiltration is better, and so on and so forth. So that's an example of how we, the pipeline essentially in the lab, and the X-ray was central to that. Without it, we really can't translate our work effectively.
Excellent. That's absolutely fascinating to me of the work that you're doing and how you're taking it from the lab to the field. Keith, anything else you'd like to add in on that?
Yes, one of the things that we've really focused on with the X-ray microscope over the past few years is trying to understand the interaction between the roots, the soil, and, as Chris mentioned earlier, the beneficial microbes. Just like with roots, previously the only way to study them was to literally rip them up out of the ground. And there are beneficial fungi that have existed in the soil for millions of years, and they've had associations with plant roots for easily that long. And the fungi will go out and scavenge in areas that the plant's roots can't get to. And they will bring those resources back to the plant. They live inside the plant roots. They will exchange these nutrients for sugars that the plant makes with photosynthesis. So it's a great mutualistic relationship. But again, these fungi exist in a 3D network out in the soil, and no one has been able to look at that 3D network without having to rip it up out of the soil or having to grow it in a really artificial petri plate in the lab. There's lots of very elegant work that's been done on that exchange, and there's a lot that's known about it from the petri plate, but no one's been able to look in 3D in the soil. And we've been able to get much closer to seeing that in a natural state. We can grow plants in a tube where they are naturally colonized by these fungi, and then we can treat that tube in such a way that the fungi are now visible to our X-ray system. So we can start to see what these networks look like in an undisturbed fashion. Certainly, again, as Chris said, we can't put an X-ray system out in the field, but what we're doing is getting closer and closer and closer to authentic situations where we can actually get important information on how these interactions happen in 3D space.
Keith, I got to ask, what did you think the possibility was that we'd actually be able to achieve imaging these microbes on roots? I remember conversations with Valent many years ago where we got a little overexcited and thought, wow, wouldn't it be incredible if we could do this? And we all thought that would be incredible, but you and I were sitting there thinking, well, no one's ever shown it in the world. I don't know. I was hopeful, I guess. Hopeful would be the word.
That's a great way to put it. I think we were both hopeful because of the unique opportunity that we had. So Valent Biosciences was certainly very interested in learning more about those microbial interactions at the root surface. And I've been an imaging guy my entire life. I spent 25 years in my previous job doing nothing but trying to find ways to look at microbes and their interactions with plants. So to have this opportunity to have this cutting edge X-ray microscope technology and the resources of soil scientists and the Danforth Center and my experience bringing all of that together, I think is the single most important aspect of being able to solve this, being given the opportunity to explore this uncharted territory, but having all the best possible tools. So yeah, it was an unknown, but I agree. I was hopeful that we'd be able to solve that.
I love it. Are you guys the only lab that has this technology?
Yes, we're the only plant science institute in the world that has an X-ray microscope dedicated to that work. There are hundreds of these types of instruments in the world, but none at a plant science center.
But I just want to comment on that because the idea is that, maybe not every, but many will eventually have this. That's our goal. It's not just to do the work ourselves. It's to raise all boats, so to speak, as a high tide. And in fact, I suggested earlier that we were the first in the U.S., at least in the public sector, where the knowable part of plant science, to have an X-ray system. But there are now multiple systems, and we have consulted, advised, and otherwise collaborated with these groups. And the X-ray microscope is sort of the next instrument that was special. But I suspect in a few years from now, this will also be at other institutions. And that's something, if nothing else, we are advocates for advocates and supporters of this type of research into the hidden half. It's actually a mission, which is to develop more sustainable, resilient agricultural systems through more effective and efficient root systems. But as part of that, it's teaching others and blazing a trail, and that's what this work's allowed us to do.
Well, I think as farmers, and as agriculturalists, I love seeing that shift to the hidden half, right? I think that's where the focus has been shifting here lately. We were so concentrated on above ground, and now finally trying to understand how do we continue to impact soil health and everything below ground. So thank you for the work that you are doing.
Yeah.
Where do you see the future heading? And what are you most excited about?
Great question. There's a few aspects to this. So I will again just say my motivation is to do the most useful, impactful thing as soon as I possibly can. So it's to take the incredible fortune we have here with a research program and working at the Danforth Center and having great collaborations, both with Valent and other industry and with academic partners, to make an impact. That's what gets me up in the morning and keeps me up late at night, frankly. And so we're working on lots of angles. I think that some of our discoveries will allow us to improve the root systems of crops. I think that we have started to be able to measure the effects of arbuscular mycorrhizal fungi and other microbes on soil and soil health. So, for example, in work where we use the X-ray, but we also use another imaging tool, which comes from the medical imaging called positron emission tomography. It's a fancy way. We just call it PET for short. But combining these two, we're able to quantify the amount of carbon that's drawn below ground by corn roots that have AMF, arbuscular mycorrhizal fungal symbionts. And we're using that in conjunction with Valent to screen better isolates and better species of arbuscular mycorrhizal fungi that continue to draw carbon below ground. And this has myriad benefits for the soil itself. And if and when we have a carbon market, should be quite valuable. Now, that's its own thorny set of issues. And obviously, there's the science part of this, there's the policy part of this, there are a lot of facets to it to make it work, but we're doing our part in the way that we, in the way that we think works best. I want to mention one other area we're working in, which is cover crops, and we've gotten very interested in cover crops in the last several years. And these are one of these things I'm sure it's quite familiar to your audience here, that on paper they make all the sense in the world in terms of basically improving the soil and adding carbon and preventing erosion and all of these ecosystem services. But we've come to learn and are actively learning what are the major pinch points and downsides, and why, practically speaking, they're not adopted over more farmland. And these are questions that can be answered through plant science, and many of them have to do with roots and their interactions with one another and with the soil. And so we've started some major research program within the lab, actually within the Danforth Center, as part of that. And we think there's a lot of promise. We've already found some fundamental, interactions that we think we can be used to provide better advice, and also to rapidly improve the traits themselves in cover crops. Some of your listeners may have heard of cover cress, which is the commercial version of field pennycress. And so we have a couple of projects on that, and we're using our X-ray system, and we're taking our basic research knowledge and capability and applying it to something that can make an impact, hopefully quite soon.
Excellent. Keith, what would you like to add to where do you see the future heading?
Well, Lisa, the future, I think, particularly for the sort of work that we're doing in Chris's lab, a lot of it depends on computational power. So I often say that I'm just the imaging guy. I get to take all the cool pictures, but there's no biology that happens until all this imaging data is then handed over to our computational scientists to start pulling valuable measurements and structural information out the other end. So we've certainly pioneered the use of X-ray imaging across scales for many different kinds of fundamental plant questions, as well as agronomy direct related questions, looking at soil structure, pore size, and aspects that are directly impacting soil health. But again, none of that provides data automatically. You have to hand all these beautiful images off to computational scientists who then can do the programming that will result in measuring and mapping pore space and pore connectivity in soil, or mapping the size and shape of different organs inside a plant. And all of those tools require, dare I say, AI. Very often folks are very frightened by what AI can do to news reports, and I think rightly so. But at the same time, artificial intelligence and various forms of that are crucial tools that we have to use in order to teach computers how to recognize the features we want them to see and then measure them.
Great point. Chris and Keith, unfortunately, our time is short and we're gotten to the end of the podcast. I have sure enjoyed the time together today, learning more about your lab and the work that you do in the Danforth Center. Thank you very much for joining us and sharing your expertise with our audience today.
It's a real pleasure. Thank you, Lisa.
Thanks, Lisa. It's a great opportunity. Thank you.
And thank you to you, our listeners, for joining us today as well. Well, this has been episode 6, the hidden half of the Sustaining Soils podcast, brought to you by Valent Biosciences. And don't forget to find us on X at Valent Biosci, LinkedIn and YouTube at Valent Biosciences, and our website at valentbiosciences.com. We look forward to you joining us for our next podcast. And if you enjoyed this podcast, please give us a 5-star rating. It helps us to reach more like-minded individuals like you. Thanks so much, and see you next time.